So we're gonna switch gears now and we're gonna talk a little bit about PFAS effect on liver disease in humans. And I'm gonna try just to give you a flavor of how we're trying to translate evidence from human studies, experimental studies into public health actions. So this is our outcome, metabolic dysfunction associated steatotic liver disease, very long title for what we used to refer as NAFLD, non-alcoholic fatty liver disease. Basically, that this is a disease that is described by fat accumulation in the liver. It affects, it's a silent epidemic. It doesn't give a lot of symptoms. It affects more than 10 million people in the US and at least one in every 10 kids. And the problem that we have with this fatty liver disease is that the traditional risk factors like genetic susceptibility and obesogenic diet or sedentary lifestyle cannot fully explain this current epidemic. So there is emerging evidence from animal studies and human studies showing that exposure to what we call endocrine disrupting chemicals, especially during critical periods of development, can predispose to a new phenotype that is called toxicant fatty liver disease. So in regards to PFAS partition to the liver, and animal studies have shown that PFAS are hepatotoxic like 40 years ago. So we know that PFAS can induce liver steatosis and can induce liver enlargement in animal studies. But human evidence from epi studies was scarce. So what we did some years ago, we tried to synthesize all evidence from human studies and animal studies in the first systematic review and meta-analysis that actually showed that long-chain PFAS or legacy PFAS exposure is associated with increased risk for liver injury. This was measured based on biomarkers in human studies, and also the same results, we found the same results in animal studies. So after this systematic review and meta-analysis, our group focused a lot on PFAS hepatotoxic effects. We published a body of evidence documenting these detrimental effects of prenatal exposure to PFAS on child liver injury, and also some recent studies on PFAS as a risk factor for hepatocellular carcinoma. And two of these papers were selected as NIH's papers of the year. So it looks like we are having a body of evidence suggesting or confirming that PFAS can be hepatotoxic. However, there are still gaps and limitations in current knowledge. We don't really know the mechanisms. I mean, we have some hypotheses for the mechanism. The models that we have available for PFAS study have their own inherent limitations. I mean, animal models cannot recapitulate human disease. Longitudinal studies, such as the studies described by Andres before, require long years of follow-up. And as of now, we don't have translational models where we can integrate studies, evidence from experimental studies into human studies and vice versa. And we're missing also the impact of PFAS withdrawal, because if with the new MCLs, we're gonna see a big change on PFAS contamination. So what is gonna happen if we reduce exposure to PFAS? So in the next few slides, I'm gonna give you a flavor of a translational research framework that we try to apply in our lab, where we start from the human studies, from longitudinal cohorts, and we evaluate the impact of PFAS over time. And then we go to experimental models with liver organoids, and we expose them to physiologically relevant doses of PFAS. And in both studies, we use multiomics and some computational toxicology. I mean, at the end, we generate big data with all these omics technologies, but the ultimate goal is to integrate this evidence from experimental studies and human studies to understand the mechanism of PFAS hepatotoxicity so that we can finally go into a solution-oriented public health action to identify the causal effect of PFAS on the target organ and then to co-design with communities interventions. So the next slides present preliminary results, unpublished results, so keep this in mind if we want to share them. And this is a collaboration with liver immunologists at the Department of Medicine, and also our postdoc, and this is a large consortium with other co-investigators. So we try to understand how PFAS alter lipid metabolism and increase the risk for steatotic fatty liver disease. So the human study comes from a multi-cohort consortium on obese adolescents undergoing bariatric surgery. And we examine in this study the effect of plasma PFAS on histologically diagnosed fatty liver disease, but also on the severity of disease. It's a unique study because we have access to liver samples. We measured PFAS both in plasma and in liver, and we also had several omics biomarkers. So these are the results on one of PFAS. It's a short-chain carboxylic PFAS PFHPA and MASLD, and it has the most strong association with fatty liver disease. You see a clear dose-response relationship with MASLD, which is the fatty liver disease. But what's interesting, because most of the human studies lack liver histopathological data, is that you see this association with the severity of disease. With MASH, which is the most severe form of the disease, is steatotic hepatitis, and also with the grade of steatosis and hepatocellular balloon. So that gives us an initial feedback that, this chemical perhaps is hepatotoxic, but how can we go now into the mechanism? So then we went to the lab and we designed these experiments to identify the hepatic biological pathways impacted by single PFAS, including PFHPA, but also PFAS mixture, commonly found in our human plasma, but also drinking water samples. And then a second aim in this experiment was to test what is going to happen if we remove PFAS from the media. You see here the design of this experimental study. So we have these 3D liver organoids that are composed primarily from hepatocytes, but also from other non-parenchymal cells. So they are exposed to what we call short-term exposure, is 7 days, or long-term exposure, which is 14 days. And then we have a third condition where we expose them to PFAS for 7 days, and then we remove PFAS from the media, and we let them recover for another week. And then the cells, the organoids are dissociated, and we perform single-cell RNA sequencing. So again, these are preliminary results from the lab. You see, these are the results from single-cell RNA-seq, and you see that spheroids exposed to 1 week or 2 weeks of PFAS showed a significant upregulation of several genes, while 1 week of PFAS removal resulted in downregulation. Then if we go more specifically to see what is happening on the pathway enrichment analysis after 1 week of PFAS exposure, you see this significant upregulation of cancer-related pathways. It's like almost the majority of pathways that came from the RNA-seq analysis, and also pathways related to fibrosis and necrosis. If we prolong now the exposure to PFAS mixture for 2 weeks, we still see the cancer-related pathways, but we see also some pathways related to cell death, to cellular stress and injury, and also to inflammatory response, as expected. And what's more interesting is this graph here that shows you what's happening when we expose the spheroids for 1 week, and then we remove PFAS and we let the spheroids recover for 1 week. So you see that the cancer pathways almost disappear from this pie, and you see more carbohydrate and lipid metabolism pathways that actually tell us that the spheroids still recover, but they go more towards what we call normalization of liver function. So that was interesting. Then we wanted to see what's happening with PFHPA, because it was the PFAS that was so strongly associated with the outcome in the human study. This is the pathway analysis on the spheroids, specifically in regards to PFHPA, and you see again that it triggers a lipid metabolism pathway, and more specifically multiple lipid oxidation cholesterol biosynthesis pathways that actually give us suggest that PFHPA can be a steatogenic chemical. So at the end, what is our goal? Our goal is to use all this information to identify people who are at risk for developing the disease. So the third step of this analysis was to integrate the multi-omic signatures from the in vitro study and the human study to identify precise risk profiles of the disease, which is steatotic fatty liver disease. So I'm not gonna go into the nitty-gritty details. There are many different platforms, web-based platforms where you can do this integrated multi-omics analysis. But basically you see what we call the metabolomic signature. The metabolomic signature is the overlapping features from the in vitro experiment and the human study. So we had 19 metabolites and 6 proteins that overlap between the 2 study designs. And what we did with these features, omic features, is that we integrate them into a statistical model developed by our colleague at USC, David Conti, which is a machine learning model for advanced risk prediction. So again, we're not gonna go into the nitty-gritty details of this graph, but you can see that basically when you incorporate all this information in the model, you get 2 groups. One group, the group with the green lines, is the group of low risk for developing the disease. And then you have the high risk group for developing the disease that is actually characterized by high exposure to the specific PFAS chemical, but also with alteration in several metabolites and proteins that were already predefined from the previous step. So, I mean, that's good, but at the end, we need to go back to the communities and we need to somehow translate these results in community-based intervention. So how can we do this? Again, this is a snapshot of what we are currently doing. We're working with environmental justice communities in East and Southeast LA. This is an area in Los Angeles that is heavily burdened by several environmental contaminants, including PFAS. Primarily Latinx population, that they live in close proximity to point sources of PFAS, and the geospatial modeling shows that these communities have higher risk of being exposed to water contaminated by the community engagement group is led by Max Ang, assistant professor at USC. We have two talented postdocs who are here with us, Liz and Sherlock, a research coordinator, and several undergraduate and postgraduate students. And the idea is that we partner with local organizations to expand PFAS water contamination measurements. We try to report back findings and data to the communities, and also we provide water filters to all participants. And I want to point out that we didn't have in our budget to provide water filters. This came from the discussion with the communities and the community orgs. They wanted to have something in return, so now all the households are getting a filter. Basically what we do, it's a thorough exposure assessment where we measure PFAS in drinking water in dust samples that we collect from the households and in plasma samples. And then we try to estimate associations between PFAS exposures and PFAS mixtures with biomarker-based outcomes that are relevant to liver injury. And again, we give to all the households these filters that were suggested by the Environmental Working Group as portable filters that can eliminate PFAS exposure. And these are just some preliminary results from the survey from the community in regards to their perception about the quality of water, and we discussed a little bit about this in the last session. 100% Latinx, majority of them from low socioeconomic backgrounds. They don't trust water, the tap water, and they don't drink their tap water. Most of them, they go and they buy water from the wateria, but they still use the tap water to cook or to shower. And when we ask about their perception about the quality of water, you see that they don't trust the water. They feel like they are dissatisfied with the color, with the taste of the water, but they say that it will be safe to drink the water if this water was clean or if we could apply some filter. So this is still to be continued because we are in the process of collecting the samples. I hope I will have more results soon. And with this last slide, I would like to thank all the collaborators at USC and other universities, and also all the community organizations that help with this project.